Today's guests are Cristina Fernandez-Valle and Houman Sadri. We join their conversation in the faculty lounge.
The muscle contracts, so the skin, so you can feel, so that the organ secretes whatever it needs to secrete. And in order for that long electrical wire to send electricity down, it needs to be insulated, like those telephone wires that have different colors on the outside. So the Schwann cell is what makes that a lipid sheath or a lipid wrapping around the axon, so that it allows electricity to go down the axon to the target organ or muscle or skin. So I study how that Schwann cell does its job.
Interesting. In some ways, it sounds like, some of the things you do are similar to some of the things I do. Basically, you're looking at these cells that they act like a means of communication. Is that right?
Yeah, this is all
between brain and certain organs.
Mm-hmm.
Interesting. Now, was this the type of research you were interested in when you were a graduate student? Can you tell me a little bit? I mean, we have talked about so many things, but I've never actually asked you about your, graduate program. Where did you do it? What did you do? And how did you come into what you're doing?
Okay, so basically, I was an undergraduate at Florida International University, which is in Miami, which is another state school that competes with UCF for a lot of the funding. And I was studying biology there with the idea of being a medical doctor. But I found that after finishing my degree and working in biomedical research, I was working at the Veterans Administration Hospital in Miami, doing work on steroids, biosynthesis, and tumors of the adrenal glands. And I would go to grand rounds in the morning, at 7 a.m. in the morning, with all the MDs and the trainees, the interns. And I found that I empathized so much with the patients that I was afraid if I made a mistake and I would hurt someone, that I couldn't sleep at night. So I decided I felt more secure in the research lab. So in the research lab, you work with test tubes full of DNA or protein. At most, you might have a laboratory mouse running around. And I just felt more comfortable there. So I developed an interest in the nervous system, because that is the central command center for the entire body. It dictates your personality, it dictates your behaviors, when you wake up, are you a night owl, or are you a morning person, and it regulates your heart. Every organ is regulated by the nervous system. So there was this particular picture in a book, I remember. It was an electron micrograph of this axon. So the axon is this round circle, and it's clear, and then it has layers and layers of this dark material around it, and that's the myelin sheath. And I looked at this picture and I said, that's an amazing structure. I wonder how that's made. So I just like to understand how things work. And I thought a clinician would tell you, these are your symptoms, you need to take an aspirin or an antibiotic. But they don't really know why you have the infection, why did that bacteria come and invade your lungs. And I was more interested in mechanisms. So I decided to go into the Ph.D. program at the University of Miami School of Medicine. So the first year actually was spent doing a lot of medical school courses with the first year medical students, because I was in a neuroscience program, and I studied cell and molecular biology, which is, my PhD is in cell and molecular biology with an emphasis in neuroscience. So you do the first year with the medical students. You do neuroanatomy, you do neurophysiology, and then you go back into the research lab and spend the next 4 years in the research lab, and taking specialized courses for graduate students, which basically retrain your brain how to think.
Interesting.
Because when you're an undergrad, they tell you what to read, you memorize, and you put it back on a test, and you get an A. Science is like, we don't know how this works. You've got to figure it out. So there's nothing to memorize. So you have to start developing new synapses in your brain so that you basically question everything and then figure out how to experimentally answer them.
You must have been very good you know, with dissecting frogs and everything when you were young? Is that right? Because I was the one who was the champion at freeing the frogs from the lab. But you must have been always good in doing that.
Well, you know, in my, you know, private, you know, all-girls Catholic high school that I went to in Miami, we didn't have that much access to dissection of animals. We might have done a frog, I don't remember. It was mostly like either worms or plants. We did a lot of plants. So, and I remember having a butterfly collection, and my sister would not let me keep my butterfly. I had this, I had caught with a net a butterfly, and I put it in a glass jar, and I was going to anesthetize it or overdose it with something so I could pin it into a board. And my sister just cried, and I had to release my butterfly. So I never collected animals. I just was not that interested in, I was always more interested in why people are sick and how to make them better.
That's interesting. Well, you know about my research that, you know, since I focus on international relations, I have to actually travel and spend a great deal of time in countries that I study. What about your research? When you conduct your research, what do you do?
Well, there is an opportunity to travel, because once you start publishing, you find that the people that understand what you're publishing are are not always, you know, they're not down the hall, and they're definitely not in Orlando. They could be anywhere in the U.S. They can be anywhere in the world. So you have a lot of opportunity to interact with people that are in Europe and that are in Russia, that are in South America, and you can start emailing, and opportunities come up. This summer I'm going to be in Italy for a conference and talking to well, they're becoming colleagues. They were scientists. They were in the U.S., now they're in Italy, and we published at the same time papers that had opposing points of view. And for a while we would like be competitors, because they thought that this molecule, this protein that allows cells to stick to their environment, to their surroundings, was important in the function of my cell that I study, which is a Schwann cell. And I would argue, no, it's this other protein. So we would have this discussion. We would meet at conferences, and we'd always say, you're wrong and I'm right. And now we've decided after 10 years that, why don't we work together? So I'm going to go give a conference in Milan in a couple weeks, and then we have written a joint grant together to the National Institutes of Health, where they're going to create some research tools that I need to use to answer my question. So I now have this expertise that's different from their expertise, but we both work on the same system, which is how the Schwann cells that are in the peripheral nervous system, how do they insulate axons so that the electrical communication can go from your brain to your toe, so you can move your toe, or you can feel that you've stepped on a nail. So both labs work on that. So, but my day-to-day life is very different than yours. Because when I go to work, it's a research laboratory.
But see, there are similarities. I was just thinking when you mentioned that you were, you know, intellectually competing with a colleague, and I like those type of colleagues when they present, you know, ideas so different from our own ideas, because that's, you know, for us that would be learning, you know. And I think, you know, you have taken obviously the natural step, the natural next step, which is a colleague that you were competing, working together on the same project. And I think this is wonderful.
Yeah, it's much more fun.
Right.
It's much more fun. And then actually, because you have both been training your brain to think about a certain problem. So all of your mental energy that we have at work or even in our sleep is focusing on the same problem. So now we have like 4 or 5 brains, and we would have never met otherwise. And yet we want to share our thinking, and then now we need someone to say, am I thinking correctly? What do you think about this problem? So we actually challenge one another, and I think at the end, when this grant gets funded, we have to resubmit it to the NIH again in, I think, July or October. We'll probably come out with fascinating new information that will help diseases that affect, like, there are certain forms of muscular dystrophy, which is a weakness of the muscle. And the protein we study is used by, it's made by neurons, the nerve cells in the body. It's made by muscle cells, and it's made by the Schwann cells that insulate and form the myelin sheath around the axon. And there's a certain part of your body called the neuromuscular junction, where the nerve and the muscle unite.
Okay.
And there's 3 cell types there. And they all use similar proteins, but those proteins allow them to carry out different functions. So when there are genetic mutations in this protein called an integrin,
Right.
either the muscle dies or becomes so weak that it can no longer supports a person's weight, so
Right.
they end up being wheelchair bound. There are other conditions where the beta-1 integrin is involved in keeping cells together so you have a good connection, and if the connection isn't tight, this myelin sheath, which is really a spiral wrap around the axon, it starts to unravel.
Right.
And then the electricity doesn't go down the axon, it gets short-circuited. So you have no control of that muscle.
Right.
So that muscle might be alive, but now the electrical information didn't get to it. So there's a lot of different physiological repercussions to having one simple protein have a mutation.
So in some ways, you're actually studying disorder within a cell. Is that right?
Exactly.
Now, if you study disorder, then could you tell me in in terms of I would understand,
That's right.
for example, what causes cancer? I mean, because what I heard, and I don't know how much of that is correct, is cancer cells are actually our own cells that they, somehow become cancer cells. So something obviously happened in our own cells that they change. So what happens?
So the theory about cancer biology, you're totally correct. Cancer cells are our own cells that are now misbehaving, and instead of following the orders and following instructions, they decide that they're not listening to anyone. They're going to do whatever they want, and what they do is they start to make too many copies of themselves. They start to divide, and one cell becomes 2, those 2 cells become 4, become 16, and then you have a tumor. And the tumor can stay in the tissue it's at, or it can metastasize, so cells will start leaving the tumor, enter the bloodstream, and they travel to distant sites. So you can have a tumor in the colon that goes to the brain or the bone, and that's when you get really metastatic terminal cancer, and usually that just destroys the function of the tissues and the human being. So what cancer theory is that some people are born with genetic mistakes in their DNA. So the DNA is the code for you. It only has 4 letters in this genetic code, and it can make you, and it can make me. So what it needs to do, these 4 codes are linked together over and over and over again, and they specify how proteins are made in your body and in each cell.
I see.
So every cell has the same genetic information, but every cell is only going to exercise some of that genetic information to become a nerve cell or a liver cell or a lung cell. So sometimes if you have a predisposition to a specific type of cancer that runs in your family, it's because your genetic code has an error in it. And that doesn't mean you will get the cancer. If you have breast cancer that runs in your family, you might have a mutation in a protein that can lead to cancer. But it may stay silent your whole life. But as you live, you're exposed to environmental insults, like chemicals that we spray on our yard to make our grass look really green, or the insecticide we put on our rose bush, or the chemicals we eat, and the sunlight that's involved in skin cancer. So all of these different environmental triggers another mutation. So now instead of having 1 mutation, you have 2 mutations. As the older you get, you end up with 3, 4, 5 different mutations, and they can be in different proteins. And each protein acts as a policeman. So they're sitting there, they're the policeman of the cell, and they're making sure that the cell behaves a certain way. If the mutations kill off your police force, then you have a cell that can divide and become a cancerous cell, because your cell has safety nets built in, so many different safety nets. And what we find is the mutations that cause cancer fell into one of those policeman proteins and knocked out 2 or 3 different safety nets, control systems, and that's when you get a cancer.
That's interesting. So, in other words, there are 2 sets of variables or factors that they can cause a law-abiding cell, for lack of a better term,
Right.
to become a rebel cancer cell. Some of them are basically, led by our nature or what we inherit, and some of them are by our nurture or our environment. Is that right?
That's absolutely right.
That's interesting. That's very interesting. Again, I see some similarity between what you do and what I do. You study, in some ways, rebel cells. I study rebel individuals.
That's true.
What about, for example, when we talk about current news, if you recall, a couple of days ago, they talked about finding some sarin gas, nerve gas, in Iraq, in one of those roadside bombs. What is a nerve gas, and what is sarin? What does it do?
Right.
I mean, how does it kill?
Yeah, so sarin was the same, I think, nerve gas that was found in, was it a Tokyo subway years ago?
I think so, right.
Yeah, that a number of individuals killed. So nerve gas is a compound that affects a specific enzyme in your body.
Okay.
And this enzyme is called acetylcholinesterase. And I happen to have done my Ph.D., spent 5 years of my life studying acetylcholinesterase. So what this enzyme does is that it terminates neurotransmission in a synapse. So for me to breathe,
Right.
my diaphragm muscle assists in breathing.
Right.
And the information to breathe is transmitted down a nerve to a synapse where the nerve, with the same neuromuscular junction where the nerve and the muscle combine. There's a molecule called acetylcholine that is secreted by the nerve to the muscle, and it makes the muscle contract. And there's an enzyme called acetylcholinesterase that is supposed to break down the acetylcholine so you don't overstimulate the muscle. And what happens is that nerve gas inactivates that enzyme. So now what you have is a buildup of neurotransmitter at the junction. So the muscle is getting way too much information.
Right.
And it actually paralyzes the muscle.
Interesting.
And the muscle stops responding. So your diaphragm muscle stops moving, so you basically asphyxiate.
So in other words, this agent, this nerve gas, somehow gets involved in the communication between brain and muscles that we use to breathe.
Right.
So if it, it changes that, or it breaks down somehow that communication, or as you said,
Right.
overwhelmed with information, and then the muscles, they stop, doing what they're supposed to. So the individual actually suffocate. Is that right?
Suffocates.
Oh my gosh.
Yeah.
Well, I hope they don't find any more of that type of gas.
I know, and it's unfortunate, but nerve gas, and there's a lot of poisonous venomous snakes.
Right.
They target the venoms that they secrete when they bite you, and the reason you die is the same thing. They target the same system, that same communication system between the nerve
Right.
and the muscle, and will actually paralyze muscle.
Wow, that's interesting.
Yeah.
Now, in what direction is your research moving now?
Okay, so right now since 1989, when I finished my PhD, what we need to do as scientists, as molecular biologists, is that we need to go into another level of training. It's very
Right.
rare when you finish a PhD and go right into faculty position. We have to do what we call postdoctoral fellowship or postdoctoral training. So you have a PhD, you go change your laboratory, and you work on a different problem usually than your PhD, and you can spend anywhere from 3 years to 10 years in this pure research environment where you're really honing your skills, training your brain to think the way it needs to think, and then you're ready to go find a job as a faculty member. So from 89 to about 95, when I came here in 96, I worked as a postdoctoral fellow and as a 100% research scientist at the Miami Project to Cure Paralysis, which was a spinal cord injury group that started in Miami due to the efforts of a number of people. One was a neurosurgeon who got tired of treating spinal cord injury patients and seeing them end up in wheelchairs. And one was Nick Bonacotti, who's a Miami Dolphin football player whose son went to Clemson, or he was in college football, And he suffered a spinal cord injury that left him a quadriplegic in a wheelchair that he moves around by blowing through a little tube.
Right.
And he has enough control of his neck to blow into the tube, and he can move his wheelchair around.
I see.
So they got together and started fundraising seriously and hiring a group to specialize in spinal cord injury. So that happened a couple years before I finished my Ph.D. in Miami, and then when I graduated, they recruited a new faculty member from Washington University in St. Louis to lead the second phase of the research. They changed directorships. The director went back to Sweden, the original director. So I joined that group, and I was studying Schwann cells, which is a really interesting cell because when you have injury to your nervous system out in your periphery, in your limbs, you can injure a nerve, and the axons that are the individual nerve fibers that are in the nerve, so the nerve is like a bundle of axons. So you take all these telephone wires and put them together, the whole large structure is the nerve. So when you injure that nerve in your finger and your arm, it'll grow back. And one of the reasons it grows back and reconnects so you can regain movement of your extremity, of your muscles, is because Schwann cells, my favorite cell, is there. And they are very responsive to their environment. They will do whatever the environment tells them. If they're in contact with a mature axon, they will wrap that axon, make a myelin sheath, and allow the electricity to go down to the muscle.
So they act like a repairman?
Yeah, they're repairmen. If you injure the axon, the Schwann cell goes, injury, injury, danger, and it de-differentiates. It stops making myelin.
Right.
It goes back to a very developmental stage, divides and makes lots of these Schwann cells. And the Schwann cells secrete molecules that entice the axons to grow back. So the Schwann cells start making food for the injured axons and nurse the axons back to health and show them the way back to the tip of your finger where you had the injury. And the axons grow back. And when the axon finishes growing from where it got injured, if you had a wrist injury, it'll dive, all of the nerve along here will dive back. And then when the injury occurs, the Schwann cells make lots of copies of themselves, start secreting the food, the axon grows right back.
Interesting.
And then once it's there, the Schwann cell says, oh, we're done growing. Now it's time for me to myelinate. And it myelinates again the axon, so the electricity goes down. Because an axon that doesn't have insulation doesn't work. That's what happens in multiple sclerosis. Different areas of neurons that are in different areas of your brain lose their insulation, and you end up with abnormal sensation in your extremities, and you can end up with muscle weakness because you lose the myelin sheath that's in the neurons in your spinal cord and in your brain.
Right. Now, your favorite cells, can we find them all over our bodies, or are there more in certain areas?
Yeah, that's a great question. These cells are only made in peripheral nerves. So your peripheral nerves exit the spinal cord, which is surrounded by the vertebral column. So then as the axons exit your spinal cord, that's where Schwann cells start to live. And they'll live all along the peripheral nerves. But they're only there. And what's interesting about them is that they have the ability to nurture or nurse neurons back to health.
Right.
In the central nervous system, which is your brain and spinal cord, that's why spinal cord injuries and head trauma is so damaging. You don't always recover function because the cells that are there don't have the ability to nurse neurons back to health. So there's a large group of people that want to put Schwann cells into spinal cord injury or into areas where multiple sclerosis patients have lost their myelin insulation, and they want to transplant these cells into those different areas and see if we can get those Schwann cells that are transplanted into foreign environments to nurse those neurons back to health.
Yeah. If you remember a few years ago, we were so sad to hear about what happened to Christopher Reeves. I mean, Superman, man who could do anything, and he hurt himself, right? What you're studying is exactly like what happened to him.
Exactly.
Is that right?
In fact, I was at the Miami Project as a senior researcher there when that happened. And he was treated in one of the best places in the States for injury, which is in Virginia, which wasn't far where he had a spinal cord injury. And what he did is he severed the spinal cord right here at the base of his spine, where the spine fuses with the head.
Right.
And it left him a quadriplegic. And he now has the Christopher Reeves Foundation, which has generated a lot of money. He fundraises all the time now. That's become his reason to live. And I've met him and I've had listened to many interviews by him. And he really didn't think he had a reason to live. His injury was so severe.
I see.
But now he's has the foundation. He funds a lot of scientists. And what was really interesting is that there was dogma. Talk about ideas. We were talking about ideas earlier, how you have to replace an idea with another idea.
Right.
When we learn in science is that a nerve cell never grows back. So you're born with so many nerve cells.
Right.
At the end of puberty, you start losing nerve cells. And that's why we age. That's why we lose our brain. That's why we have Alzheimer's. Is once you lose a nerve cell, they're gone forever. And the dogma was that spinal cord injury had killed neurons, and there was no way you'd ever get better. So there was no research in the area because all the scientists were being taught, these fresh minds that come to college and they want to learn, they're being told, don't study spinal cord injury or brain injury because there's no way you can regrow back a neuron. So that was the dogma. And then during the time just preceding Christopher Reeves and after Christopher Reeves' injury, he has funded so much research into the area and has refused to believe that there's nothing that can be done about this loss of neurons, that he now has, helped to change the way we teach neuroscience in college. We say that you can grow new neurons. We don't know how to do it, but it can happen, and there's examples of neurons actually being born in the brain of old people.
That's interesting. So in other words, by just surviving, is that right? This in itself is a success story.
Yeah,
is that right? It's like a miracle, or that's
and actually surviving and having the money to treat himself, to go through physical therapy. Because when you had a patient who had spinal cord injury, the doctor would say, I'm sorry, you're in your wheelchair for life. There's nothing we can do about it. Learn to live with it. Go to occupational therapy. Now they say, no, there's things you can do. Exercise. They can put electrical stimulators on the muscle to keep the muscles contracting artificially so that if the nerves do grow back and the electricity is restored, you have a muscle that's receptive. There's a lot of physical rehab